So good afternoon and welcome to the NIH Director Wednesday afternoon lecture series. My name is András Orosz and I am the co-chair of the proteostasis scientific interest group with Len Eckers, and we are the hosts for today's speaker. And it is my great pleasure to introduce today's speaker, Dr. Peter Walter, who is currently an HHMI investigator and a professor and former chair of the Department of Biochemistry and Biophysics at the University of California, San Francisco. Peter was born in West Berlin, Germany, and the path toward his success began in the 1960s with the curious tinkering with chemicals in his parents' drugstore as a young boy. Continuing on this trajectory, he earned an undergraduate degree at the Free University of Berlin in chemistry and a master's degree at Vanderbilt University and a Ph.D. in biochemistry from Rockefeller University. As a graduate student, he explored how proteins know where to go, and what he found, the molecular machinery known as the signal recognition particle that brings nascent linear proteins into the folding factory of the ER, propelled his advisor, Günter Blauber, to the 1999 Nobel Prize in Physiology or Medicine. Dr. Walter moved to from the Rockefeller to start his own laboratory at UCSF, where he embarked on his lifelong journey of curiosity-driven research with a brand new focus, searching for the signals that tell the nucleus where the cell's protein folding factory, the ER, is overloaded. His group identified an ER-resident transmembrane kinase, endoribonuclease. It's called IRE1, one of the three sensors of the folding capacity within the ER lumen. And which is initiating a signaling pathway known as an unfolding protein response. The serendipitous discovery of the UPR in yeast is one of the shining examples how basic research may lead to outcomes which are relevant to humans and human diseases, since protein misfolding is a common denominator in a broad variety of chronic non-communicable diseases. During the last 2 decades, his pioneering research of the unfolding protein response earned him a long and impressive list of accolades, including the Shaw Prize and the Lasker Award in 2014 and the Vilcek Prize in Biomedical Science in 2015. He is also an elected member of the National Academy of Sciences. Outside of the lab, Peter is an accomplished woodworker and welder, creating whimsical garden sculptures and wooden clocks and fountains. He is a highly successful mentor and proponent of open access publishing and the past president of the American Society of Cell Biology. Lately, his lab identifies small molecules that modulate the activity of each UPR branch in mammalians in the hope that one day tweaking the system will help patients. So he will describe his unconventional path to discovery in today's seminar entitled From Protein Folding to Cognition, a Serendipitous Path to Discovery. Please join me to welcome Dr. Walter at NIH. I also want to mention, before everyone's run away at the end of the seminar, that there will be a reception sponsored by the FAES in the library right after the seminar.
Thank you so much. Very, very warm words. It's great to be back here and be invited to this to give this very prestigious lecture. So thanks for all of you for showing up. Despite the fact that you could also see it on a screen in the comforts of your office. So what I would like to do today is tell you a story that is sort of reflective of the fact that discovery, the path to discovery is not linear, it's meandering. And I think this is one of the wonderful things of our careers, is that we get constantly exposed to new things. And for me, it is this adventure of suddenly ending up with something you had no idea that you would ever go there, that makes our profession so rewarding. And then, as you'll see in this talk here, we are going to go all the way from protein folding to the process of cognition. And that puts us in positions where often we study things we have no idea yet about, and we have to learn new things, right? Or we can tell others that it's really not my area, so maybe we just pass it on to somebody else. But I find this opportunity to constantly learn new things incredibly refreshing. So what I want to do is tell you a little bit about the basics of how we started, and so you all can follow, bring you up to the latest, how we think about this process. But I would also like to portray to you a little bit on this path to discovery as I go along, and how we got where we are. And I gave this talk a subtitle, and that's how a phone call can change your day. And somewhere in the middle of the talk, this will make some sense. So the story starts in the endoplasmic reticulum, which is the compartment where proteins that eventually end up on the cell surface begin their life. That's where they get folded, assembled from subunits, modified. And then they get packaged into vesicles, moved down the secretory pathway to be displayed as membrane proteins or secreted proteins on the cell surface. And this movement through the secretory pathway is a step of quality control for the fidelity of the protein that the cell displays on the surface to communicate with its environment. And it's an incredibly stringent quality control. Some of the important signaling molecules, T-cell receptor, acetylcholine receptor, cystic fibrosis conductance channel, the vast majority of the polypeptide chains that are being synthesized never leave the endoplasmic reticulum, but never make it to the cell surface, but rather are degraded. This is, of course, an enormous energy expense for the cell, but I think it illustrates beautifully that what's really valued is the quality, the fidelity of the proteins, rather than conserving energy. And in this is reflected in the fact that if you look at a cell, the professional secretory cell, in this case, the exocrine pancreatic cell, in this beautiful electromicrograph from George Pallades, that this cell is filled wall to wall with the compartment that makes this folding possible, the endoplasmic reticulum. The cell makes its own weight every day in secretory proteins, so this goes hand in hand, the specialization goes hand in hand with the amplification of the structure responsible for assembling them. So how does a cell know how much endoplasmic reticulum it needs? You could think about that in principle in two different ways, one of which is that the cell in development receives a signal that tells that you're going to become a secretory cell, and the cell responds by building the appropriate machinery to do so. Or it could be that the cell receives signals that increase the production of secretory proteins. They enter the endoplasmic reticulum, but reach a limit of the protein folding capacity, and that creates a feedback signal that then tells the gene expression program to make more of the organelle according to the need of the cell. And the unfolded protein response is signaling of this latter kind, where we have communication from the lumen of the endoplasmic reticulum to the gene expression program that adjusts the abundance of the endoplasmic reticulum. And it's called the unfolded protein response because it initiates by an overload of proteins entering the endoplasmic reticulum, and these proteins then, not being able to fold properly, unfolded or misfolded proteins accumulate, and they act as agonists for a set of receptor molecules, sensors, that monitor the conditions inside the ER and transmit that information across the bilayer of the membrane. In metazoan cells, there are three principal branches of this signaling network. Each of these leads to the production of characteristic transcription activators that then activate downstream UPR target genes. And these transcription factors are produced by the activation of these sensors by very unconventional signaling pathways. IAE1, as you already heard from Andres in the introduction, uses a very unique mechanism of regulated mRNA splicing that we discovered in yeast, but that is all this, the principles are true in mammalian cells also. ATF6 is a transcription factor that, very similar to Brown and Goldstein's cholesterol biosynthesis transcription factor, SREBP, is initially made as a transmembrane protein, and then when unfolded proteins accumulate in the endoplasmic reticulum, it is moved by a mechanism that we don't understand at all currently to the Golgi apparatus, where it meets up with two proteases that release this fragment, which now, severed from the membrane, can go to the nucleus and be the transcription factor to collaborate with these other guys. And finally, PERK in the middle is a transmembrane kinase that uses a mechanism of translational control to produce this transcription factor ATF4. And the goal of activating these pathways is to bring the system, which has an imbalance between the load and capacity of the folding machinery in the ER, back into homeostasis, so to adapt. If this cannot be achieved, however, then the cell makes an active decision to undergo an apoptotic cell fate. So rather than producing potentially misfolded and malfunctioning proteins and putting them on the cell surface, the entire cell decides to remove itself from the organism. And it's this decision then that the unfolded protein response makes for cells, a life-death decision, that puts it in the center of many different human diseases. Maybe the most intuitive ones are where we have familial mutations, like in retinitis pigmentosa, where rhodopsin molecules are encoded with a mutation. And when these proteins enter the endoplasmic reticulum, not only do they not work properly, but the cell gets frustrated by having these misfolded proteins it cannot do anything about because of the mutation, and the cell kills itself. So the entire retina in this inherited form of blindness degenerates by apoptotic cell death. Similarly, in type 2 diabetes, the beta cells in our pancreas are asked to make ever-increasing amounts of insulin, eventually exceeding the protein folding capacity in the endoplasmic reticulum, and the cells die of apoptotic cell death. And of course, if one cell dies, the cell next door is asked to make even more insulin, so it becomes this degenerative spiral. But seemingly contradictory, in other instances, the unfolded protein response can also keep cells alive that we would rather have die. And examples of that are virus infections, where cells use this pathway to make more endoplasmic reticulum, to envelope the viruses, to make enough membrane proteins for that. So if we could develop some pharmaceutical that would bias this life-death decision, we could potentially make very effective antivirals from which the virus could not mutate away, because it's a cellular function that we're affecting here. And then we have cancer, where all cancers have in common the fact that they evolve to then grow in our body in places where they should not grow. And this mutational process that takes place in them caused and leading to increasing genomic instability leads to many mutations that accumulate, not only the driver mutations, but many passenger mutations, because evolution isn't directed. And so the proteome in these cells has many mistakes, and these pathways, not just the unfolded protein response, but also heat shock pathways, are activated in these cells and provide protective mechanisms. And we are engaged in a nice collaboration right now with Genentech to actually explore different virtues of these pathways in whether we can manipulate them in a cancer setting. But as I just told you, the unfolded protein response then can be both good and bad at the same time. And I think the challenge really is for us to figure out at this point whether we can come up with a therapeutic window, where by manipulating these different pathways, we can actually do more good than harm in a clinical setting. So a couple of years ago, we engaged in a project funded by Howard Hughes Medical Institute, where we screened for small molecules that would then function as tool compounds to affect these different branches of the pathway. And I'm happy to report that we now have, for each of these branch, we have both inhibitors and activators at various stages of refinement. So this project is working quite nicely. And I want to tell you today about progress we made in one of these branches, and that is the PERK pathway in the middle here. So PERK uses a mechanism of translational control that affects the production of this transcription factor, ATF4. The project I'm going to tell you about was initiated and carried in the lab by a single postdoctoral fellow, Kamila Sidrowski. So the step that is affected by PERK signaling is a very central step in translation, protein translation, that is operating in every cell and leads to translation initiation at pretty much every AUG codon in the mRNA, in the transcriptome. The EIF2 is a general transcription factor. It's a trimeric protein that exists that is a GTPase, and in its inactive state, it's bound to GDP. And then when GDP is exchanged to GTP, it becomes active, binds initiator tRNA, methionine tRNA, and then initiates translations at all AUG-initiated open reading frames. As for many GTPases, this reaction is not spontaneous, but is catalyzed by a GTP exchange factor, which is called EIF2B. But since it's such an awful nomenclature, if I talked about EIF2 and EIF2B, I'm just going to refer to it as the exchange factor, or the GEF, for this reaction. This exchange is regulated by the fact that EIF2 can become phosphorylated at a single serine residue at the alpha subunit. And it's this phosphorylation event that makes it stick more tightly to the exchange factor. The exchange factor is limiting in cells, and therefore is titrated over here. Effectively, the phosphorylation then blocks this exchange reaction from happening. Okay? So we got into this, studying the PERK pathway. So PERK phosphorylates at this serine residue here, sensing ER stress. But there are other kinases in the cell, GCN2, PKR, HRI, that sense different stresses, are activated by different conditions, such as double-stranded RNA, indicative of viral infection on this kinase here, amino acid starvation, so nutrient availability, GCN2, or the redox state indicated by some heme deficiency here by this kinase. And all these kinases come together and integrate the information at this single serine residue here where the phosphorylation takes place. And because of this integration point, this response is referred to as the integrated stress response. So it's a subnetwork of the unfolded protein response in this context. And the consequences are, as I just told you, the general inhibition of translation, but again, seemingly paradoxically, we also have the selective translation of some mRNAs, including our transcription factor ATF4 that functions in pretty much every cell, but also some selected mRNAs that are tissue specifically expressed, such as this protein OPHN1 that is neuronally expressed. How this comes about was very elegantly worked out by Alan Hinnenbusch in the yeast system initially, where we have EIF2. Translation is up if it's not phosphorylated. But then there are some mRNAs that could turn little upstream open reading frames. And when these upstream open reading frames are translated, the ribosome doesn't make it down to this AUG, so ATF4 is not produced. But when now phosphorylation sets in, Translation generally is blocked, but now ribosomes are allowed to access to this AUG residue downstream of this open reading frame, and ATF4 is produced under conditions of limited initiation capacity in the cell. So what we did, very simply, we looked for a small molecule. So we replaced the ATF4 open reading frame with luciferase, and we then created a reporter cell line and screened a small molecule library. We induce ER stress, so now luciferase is being produced under these conditions. So the light goes on, you add various compounds, the light goes off, and you have your inhibitor. That sounds very easy. It took us a couple of years to do that. And in the end, we had sorted through some 100,000 compounds, sorted them in secondary screens that they're only hitting the PERK branch of the unfolded protein response, that they are affecting the endogenous proteins, not just the reporter proteins, and so on and so forth. In the end, we ended up with one compound, and our screening facility decided to throw it away. It didn't meet their triaging criteria because it wasn't soluble enough for them to think it would ever amount to anything. It's a beautiful little compound, and we call it ISRIB. So everything else, what I'm going to tell you from now on, is basically due to the fact that Kamila Sadrowski decided to pull this compound back out of the garbage can and say, I want to study it anyway. So she decided to ignore the advice of the experts, and I'm telling you later why this may be the case. So here's a never ever think outside the box. This is sort of, she was reminiscent of a few grant reviews you got back yourself. So the compound is, since I started my career in chemistry, it's a very beautiful compound. There is beauty in symmetry. It's two-fold symmetric, so these two arms are the same. And in the middle, we have this cyclohexane ring, which can be substituted two different ways. You can have the substituents in cis or in trans. The trans compound is active. The cis compound is completely inactive. So we have virtually identical chemical matter that provides a nice control. We've made numerous mutations, some 200 derivatives of this molecule, and we've pretty much only made it worse. So right out of the screen, it seemed to be quite nice. We can make some substitutions here at this ring that make it a little bit better. And then if we do the same on the other ring, it makes it a little bit better still. If we make some substitution on this ring that make it work worse, and we do the same on the other ring, it makes it worse still. So in other words, this little compound seemed to want to tell us, I want to be two-fold symmetric. That's going to come through in the rest of the talk. So here's what it does. You take cells, you label them as S35 methionine, run the proteins out on a gel. You can see we get nice protein synthesis, no ATF4 is being produced. We now induce one of the kinases, here shown for ERS, so the PERK kinase. There's protein synthesis is severely down-regulated, and now ATF4 is being produced. You add the ISRIB to these cells, and you see that now the effect of the ER stress is entirely reversed. So it's a very effective way. There are different ways you can read out inhibition of translation initiation. Another way of doing it is to look at the mRNAs that when they're covered with ribosomes and translating actively, they are prevented from binding RNA binding proteins. Once initiation is inhibited, RNA binding proteins can hop onto them and via the low complexity sequences get dragged into these RNA granules, stress granules, which we can follow by fluorescence. You see here we have cells in which we induce ER stress. You see stress RNA granules form. And then if we do the same in the presence of the ISRIB, you see that the stress granule formation is entirely prevented. This is, we can preform the stress granules. We can then add the compound, and within 2 minutes they're gone. So these granules are really reversible storage forms in which the mRNA is put aside for a while and from which it can be retrieved incredibly fast once protein synthesis is going again. So to make a long story short, this is of course a cell-based screen. We have no idea where on the pathway does our compound act. We were most interested in the unfolded protein response, but it turns out that it does not affect PERK activity. PERK still autophosphorylates. And ERF2 is still phosphorylated. So these parts take place. It blocks the translation attenuation downstream, all of these different kinases that we've tested. And it reverses all the phosphorylation-induced translational changes that we can measure, which we tried to do very carefully with Nick Ingoglia by ribosome footprinting. So he's one of the inventors of this technique in Jonathan Weissman's lab at UCSF. And I showed you it reverses stress granule formation. So ISRIB somehow blocks downstream of this phosphorylation event. It makes cells immune to this phosphorylation. It's just as if it never happened. It's a remarkable compound. Right out of the screen, we got an in-cell efficacy of 5 nanomolar. So normally when you do these things, you're happy if you end up in the micromolar range, and then it takes a lot of chemistry to refine the affinities and to make it better. But this stuff worked at 5 nanomolar, which explains why the screening facility was triaging against it, right? Because you cannot walk very far with limited solubility. But at 5 nanomolar, you don't need much in solution. So it was perfectly fine. We made it about 10-fold better by just, as I told you, putting little substitutions on the side rings here. So we're in the high picomolar range now. Right out of the screen, it has good bioavailability if we inject it into mice. The half-time in circulation is about 8 hours. It has good blood-brain penetrance. Again, a property you normally need to work on, whereas it was just given to us from the primary screen. It is orally available in the animals. And most surprisingly, to date it has shown no overt toxicity, which is very surprising since this pathway exists in every cell in our bodies. So we have injected mice for weeks and not seen any adverse effects. So what does it bind to? So all the sexy techniques that we teach to our students, putting biotin handles on, UV cross-linking reactions, click chemistry, and so on and so forth, only pulled out crap. They did not allow us to identify the target of ISRIB. So the only clue we had that the structure-activity analyses indicated some importance of some twofold symmetry. So what is the molecular target? So the first clue came from an experiment, everything else having failed, that we did together in collaboration with Jonathan Weissman's lab, where we did an shRNA screen to basically ask which gene in the cell, when knocked down, will make the cells more or less sensitive to the action of the drug. And this screen identified 2 of the subunits of the exchange factor that impaired ISRIB action. Then at the pretty much same time, 2 papers appeared that showed that in cells, this exchange factor is not just a heteropentamer, but exists in an equilibrium with a decameric form. So a dimer of the pentamers. And of course, whenever you form a dimer of something, you have the opportunity of forming a twofold symmetric interface. So what this suggested then is a model, but maybe ISRIB binds here in the middle, and then pulls this equilibrium from this pentameric form into the decameric form. So Jordan Tsai in the lab tested this directly by putting cell extracts, treated or not treated with the drug, on sucrose gradients and just looking at the size of the complex. I love sucrose gradients. And I think this in vitro veritas here. So here's where the pentamer sediments in the gradient. Sedimentation is from left to right. These are bands, control proteins on the gradients. And if we now make a cell extract from cells that had been treated with the drug, you see that the subunits of the exchange factor shift in the gradient into the position of a decamer, whereas all the other proteins stay put on the gradient. So I think this experiment just beautifully illustrates this pentamer-decamer conversion due to the action of this drug. Okay, this of course was a biased experiment. We only see the bands we want to see by probing with the antibody on the Western blot. So we did the same experiment, but now by mass spec profiled through the peak of the decamer, and then correlated all the proteins we identify with one another. And here at the very top of this correlation stand out all the 5 subunits of the exchange factor. So what this means is that there's no other protein that hops onto this complex or provides this decamerization, but it's most likely just the pentameric form. Now, to summarize a lot of enzymological work, taking ISRIB and adding it to the GEF increases the specific activity, the exchange activity of this enzyme, by about 3 to 5-fold. So it is an enzymatic activator of this activity. And it lessens, but does not block completely, the inhibition by limiting amounts of the phosphorylated form of the ERF2P. And I think what this says, so if you add enough of the inhibitor, you can still block the exchange activity entirely. And I think what this may tell us is it provides a nice explanation why this compound is not toxic in cells, that at high concentrations of the inhibitor, so when the unfolded protein response is fully turned on, this pathway would still work, and it would still provide the protective effects of the unfolded protein response. But what we have is we have now a different dose-response curve. Probably due to dimerization, we can put in some allosteric regulation, not just needing one, but now needing 2 inhibitors, so changing hyperbolic inhibition, Michaelis-Menten condition to a sigmoidal curve. And that would allow cells then to regulate something in the middle, but keep the protective effects intact at the end. So we are pretty sure that ERF2, that the exchange factor, is the target of the drug. And that was independently validated by our sometimes collaborator, sometimes competitor, David Ron, who identified somatic cell mutations that make cells insensitive to the actions of ISRIB. And these mutations mapped into one of the subunits of the exchange factor. Well, seeing is believing. 2 fantastic graduate students in my lab set out and purified the exchange factor and ERF2, which we can now have recombinantly in large amounts. Just to give you an example, if we pull out the exchange factor from HeLa cells, we get about 5 nanograms from a liter of cell culture. This is a recombinant system. In E. coli, we get milligrams. So it was really necessary to produce these proteins, which was very difficult. And the trick really was we have to express all 5 subunits in the same cell in order to get the particle assembled. If we try to express them separately, we could never put it together. With that material in hand, we collaborate with Lakshmi and Adam Frost, and we have, by cryo-EM now, a structure, the first structure of a human ERF2B, the exchange factor. And you can see in the shape here, it looks very much like a crystal structure that was previously determined in yeast in S. Pombe. This is what the structure looks like in higher resolution now. We get it at 3.4 angstrom. And there in the middle is our drug. This is photoshopped. But it's just, the technique is absolutely amazing, what you can do. and the density fits perfectly, and we can trace the chains based on the analogy to the Pombe structure. So we now have a very nice molecular model of the binding site of the drug, smack in the center, bridging two of the heteropentamers, just like we've drawn in the cartoons before. And these residues indicated here are the residues where David Ron's mutations map that make EIF2 insensitive to the ISRIB. So this is, all of this is basically to tell you we know a lot of how this thing works. It's very high affinity, binds in the middle, and the dots connect beautifully. So somewhere during this work, I called up a colleague of mine, Nahum Sonnenberg at McGill, and the purpose of the phone call was basically to ask him if he has anything. He has been studying translation initiation for his career, whether there's anything in the freezer that would be useful for us, whether we could exchange some reagents. And he offered everything he had, but he didn't have very much, so this wasn't very useful. But what he pointed us to is results from his lab in which he had linked this very pathway of the integrated stress response to the process of memory consolidation. It's an old concept, right? Goes all the way back to Eric Kandel, that learning in aplesia requires ongoing protein synthesis and then affects synaptic plasticity. And results from his lab, the experiments, were that if you take a heterozygous mouse in which the single phosphorylation site in EIF2 is changed to from the serine to an alanine, so it can no longer be phosphorylated, then these mice learn better. So the homozygous mouse is dead, but it doesn't develop because of pancreatic failure. But the heterozygous mouse, which can only make half as much of the phosphorylated form of EIF2, is perfectly fine, and it learns significantly better. So we send him some of our drug, which affects this very same pathway that his mutational and genetic analyses pinned down, and he put it into their behavioral assays. And I think the important thing here is that at this point in the story, we didn't know that this compound had any bioavailability, or that it would cross the blood-brain barrier, and they were just adventurous enough to do the experiment anyway. So here is the results are amazing. So we tested this in this Morris water maze, where you basically the mouse is put into this milky solution, has to find this hidden platform. It orients itself by visual clues around the room, very much the same way we would orient ourselves in a building. And then you just measure the time it takes for it to find the hidden platform. And in this light learning regime, where you train the mouse lightly enough that we still have room for improvement under the curve, they plateau at this performance level. But if they have been injected with the drug, you see that they learn substantially better. And these curves basically parallel the curves from the heterozygous compared to the wild-type mouse. We can use different assays, such as contextual fear learning, where the mouse associates this particular environment with an electric foot shock, and next time it encounters this environment, it starts freezing, and you measure the freezing ability of the mouse. Having the drug injected makes no difference in the short-term memory, but after 24 hours, so when memories are moved from short-term memory into long-term storage, you see that we get a significant difference. And in this case, the drug was injected after the training period. So in other words, it's not perception, but it's really the movement of short-term memory into long-term memory. And the same in auditory fear learning in rats. In this case, the animal associates a nasty sound with a foot shock. Again, no difference between drug-treated animals or control animals in a short-term regime, but after 24 hours, animals that had been injected learn significantly better. So what we have done here then is we've injected this compound into a wild-type animal, and we have improved their performance in these assays. So what this tells us is that the integrated stress response via this phosphorylation event must inherently limit memory consolidation. So there's a break on, and with the compound or the genetic manipulation, we can release that break. So there's extra capacity to be tacked into. And I find that very fascinating, right? Because it basically means that evolution hasn't optimized this process, but rather, and they're probably for good reasons, which we can come back to in the discussion, but there is basically a reservoir which we can tap into. So how could that be the case? Well, you all know that memory consolidation and learning is associated with changing the strength of synaptic transmission, having post-synaptic glutamate-gated channels, the AMPA receptors. And the strengths can change in two different ways. We can have long-term potentiation, which strengthens the synapse by increasing the number of AMPA receptors, or we can have the reverse, long-term depression, which weakens the synapse by decreasing the concentration of AMPA receptors. So we collaborated with Mauro Costamaggioli at Baylor, and Mauro is actually the postdoc who brought these behavioral studies into Nahum Sonnenberg's lab, and now has his own lab at Baylor. And he has found that in the genetic model that he had developed, it is the process of long-term depression that is being affected by this phosphorylation event. And what you're seeing here is a synapse that's being stimulated and recorded from. When you treat it pharmacologically to induce this process of long-term depression, you'll see that the currents go down and then don't recover to the same level, which is due to the fact that the concentration of AMPA receptors has been reduced now. This is what happens, this is a control, this is what happens in wild-type animals. If you do the same in the heterozygous animal, you see that this weakening of the synaptic connection is entirely blocked. So in other words, the synapse stays stronger. We can do the same with a drug and with the ISRIB, and we get parallel results. So again, long-term depression is blocked under these conditions. We can look more directly at post-translational AMPA receptors by immunostaining of these synapses. So you start out at 1 here. You then, if I have a wild-type animal, you again pharmacologically induce long-term depression. So the removal of AMPA receptors by endocytosis, you have about 40% left after you're done with this. If you do the same in the heterozygous animal, this reduction of AMPA receptors does not take place. You can do the same with our drugs. You start out at 1, you induce long-term depression, you go down to 40%, you do it in the presence of the drug, no reduction takes place. You do it with an inactive analog of the drug, nothing happens. So what we have here is we have a perfect match between the genetics and the pharmacology of this little compound, which gives us, I think, great confidence that this stuff is actually acting on the target that we assume. So Mauro went one step further, and he identified in neurons an mRNA encoding the protein OPHN1, which is neuronally expressed and is now translationally upregulated in the process of long-term depression, and was previously implied to be involved in the process of AMPA receptor downregulation by the endocytic event. So it's a Rho-GTPase modulator. And this mRNA contains the same little open reading frames as the ATF4 mRNA. So it's under identical or similar translational control as we know for our transcription factor in the unfolded protein response. He's gone one step further and added shRNA to this protein and shown that we can block this reaction, the downregulation of the receptors entirely. So what we think then is that the action of the drug is by preventing these actions downstream. So it's a reasonable working hypothesis, I think, that allows us to connect the dots. So what do we do with this? And I was really, really amazed how quickly one can slip from curiosity-driven basic research into issues that are actually ethically quite complicated. We've identified a cognitive enhancer, and of course we don't want it to fall into the hands of the tiger moms who give it to their daughters the day before the exam so they can perform better. But look for meaningful clinical applications. So here, ISRIB, the taste kids love. Could we please have some more? The most immediate clinical connection is probably a disease called vanishing white matter disease, in which it's autosomal recessive, so the children inherit partially defective alleles from both parents. And then it leads to severe neurological degeneration and cognitive impairment. And 95% of the patients in this disease have mutations in the exchange factor EIF2B. And these mutations splatter over all of the subunits of this exchange factor. So they're most likely hypomorphic mutations that reduce the activity of this enzyme and therefore lead to various degrees of clinical consequences. But here we have an enzyme activator, right? So I think we have a pretty good chance of perhaps making a difference to these patients. It's a very rare disease, but these kids currently don't have any hope. So this might be a nice indication. And Kamala Sadrowski has since moved on to Calico, and her project is to build a mouse model and to test the usefulness. There are many other cognitive functions, cognitive loss of cognition models. All the neurodegenerative diseases you can think of fall into this category. We, of course, have no hope here to cure these diseases, which have very different origin, but maybe we can make the life of the patients a little bit better by tapping into this extra reservoir of cognition that may be out there. And finally, as is becoming increasingly apparent to me, we also have decreased cognition due to normal aging, so hopefully this will move fast enough for me. In all of these models, however, we would need to give this compound chronically. There are also acute cognition models. One is postoperative cognitive decline, where after severe surgery, like hip replacements, you initiate, due to the tissue trauma, a cytokine storm that leads to brain inflammation. And the next day, you're not going to be quite as smart. And for most patients, that makes no difference. It just goes away. It cures itself. But in particular, for elderly patients, it can lead to permanent dementia. And maybe we can prevent something there. And then we have traumatic brain injury models where mechanical insult to the brain leads to inflammation and cognitive loss. And I want to close this talk by showing you results in this latter model of traumatic brain injury here. And we have collaborated with Susanna Rossi at the Trauma Center at UCSF. And Susanna has built this model where you inflict mechanical insult, controlled mechanical insult to the brain of a mouse, and you then test these mice in these behavioral assays. So in this case here we have a similar test as I showed you before in cartoon version, but now we have these radial arms here. This is a control mouse. It has learned to find this hidden platform over here, again orienting itself by visual clues around the room. And down here is a traumatic brain injury mouse, which basically swims in a random pattern. It doesn't learn. And this movie goes on forever. So we'll drop it short. Experimental design of the data I'm going to show you is that we inflict the injury. In this case, it's a direct insult, a contusion on the brain at this point in time. We then let the animals recover for 4 weeks. So after 4 weeks, it's the first time they see the ISRIB injection. They get trained the next day. They get injected again in the evening, trained 1 more day, injected again, and then the next day we perform a memory test. And then no more injection, and a week later, another memory test. This is what the data look like. No drug in this experiment yet. So here are the animals learning how to make fewer and fewer errors in this. This is the day 1, so 4 weeks after the injury. And the animals that control mice, and these are the mice that received the injury. And as you see, they don't get any better. Control mice, the next day, next block of learning, they remember what they've done the day before, and then they get better and better again until finally they make hardly any mistake anymore. A day later they remember, and then a week later they start slowly forgetting, but so would we. And this is the data with the drug injection. So again, superimposed here, we inject the ISRIB to the injured animals. So already on the first day, you now have a positive slope. They start learning something. In this case here, there's no difference between the control mice or control mice injected with the drug, because with the strong training regime, there's no room down here to improve much. And then the next day, the magic happens. I mean, to me, this is absolutely magical. The injured mice, the red dashed line here, now become indistinguishable from the control group. A day later, they remember. A week later, they remember. So this is still small numbers of animals, but I think looks incredibly promising. And the statistics are quite nice. So this is how the individual animals performed in this assay. Even though it's not statistically significant, I think you see a slight trend here that the drug-injected control mice also improve their learning and make fewer errors. Okay, so we tried to make it, the last data piece here, we tried to make it a little bit more difficult for them. And this is another memory test where we have a table with some 40 holes, and the mouse has to remember only one of the holes has a little escape hatch underneath, so they don't have the stress of swimming. They're just being put in the middle. They don't like to be exposed to bright light, so they find a cozy place to hide over here. Again, using visual clues around the room, the TBI mouse basically walks a random path to find this hole. And then the next day, we do the same test again, but now we change the position of the escape hatch. So the mouse has to forget what it learned before, has to relearn the task, but at the same time, it also learns how to learn. So it gets faster and faster in doing that. So it's like you're finding your car in the parking garage, right? The garage is always the same, but your car's in a different spot. And admittedly, my path looks like this. And here are the data. We do this again, very similar protocol. So we're a couple of weeks after the injury, and you see that by day 3 here of this learning paradigm, the injured animals here without the drug stay flat, whereas the injured animal that received the injection now start learning and remembering the platform, eventually becoming statistically indistinguishable from the control groups. And here you also see that for the control group, that's a drug injection, that they reach this level of maximum performance already on day 2, so much earlier than if they didn't receive the drug. So we have this improvement on the uninjured animals over here. So this is where we are. This is good news for the mice. We have, of course, no idea whether any of this will translate into a human situation. Mouse brains and human brains are quite different, and many studies on TBI have failed before. But we are very optimistic, and definitely will pursue. So here's the attitude of the people in my lab. Never mind what's holding it up. Keep hammering. This is my role here. And these are the students and postdocs. And it's not just bringing in the material and to write the grants and stuff, but it's also to project confidence that eventually we'll reach terra firma again over here. Let me summarize what I told you today is that we've identified this beautiful little molecule here as a potent and first drug-like molecule to block the integrative stress response. That ISRIB binds to and activates eIF2B by dimerization, and we now have a very clear definition of the binding site, which brings up the question, does a binding site like that evolve for a synthetic compound like that, or is there normally some metabolite or some cellular constituent that binds there and affects this dimerization reaction in a physiological setting? We've shown that ISRIB's efficacy has efficacy as a cognitive enhancer. And with that, we have identified the integrated stress response as a druggable pathway and its inhibition as a promising avenue to combat cognitive disorders. It has an amazing efficacy in the models of the traumatic brain injury and data I'm not going to show you, but we have similarly promising results for the post-operative cognitive decline assays done with Mervyn Mays at UCSF. And what I'm most proud of is that what Andres already alluded to in the introduction is that we had no idea we were going there. Right? You could have never written a grant on a project like that and said we're going to study neurocognition. But that all of this emerged by walking down this meandering path with curiosity-driven basic research. And I think we are now at a stage where we are translating things, but we definitely didn't set out with the idea of knowing what might be translatable when we got there. Based on this, the magnitude of the different techniques and expertise that were required, this has been a massively collaborative investigation, which, as I told you, has benefited greatly from open communication. And these are the people in my lab. Carmela now moved to a position at Calico. We licensed the drug to this company, and hopefully they will put it into clinical trials. So all of these people here contributed in very major way to what we've accomplished, and thanks to Nahum, Maho, and many others. And with that, thank you. I'm happy to answer questions. You want to?
I was wondering whether the effects of the ISRIB in the TBI system are also linked to changes in the levels of AMPA receptors, as you showed with synaptic plasticity.
Yeah, we want to do that. We don't know that yet. It's also, we are looking at we need good readouts, right, for target engagement, like AMPA receptor or the spine dynamics. So many of this work is ongoing. We don't know yet.
Congratulations for an exciting work. We definitely need more drug molecules for neurodegeneration and all the other issues. So looking at this structure, I see you have two peptide bonds. So are they susceptible to degradation after you mentioned intraperitoneal injection, but ultimately they have to end up in the plasma, I guess. And
yeah. Are the peptide bonds hydrolyzed? We know nothing about catabolism or metabolism of the drug at present.
And how about the partition coefficient? If they have to go to the brain, obviously you need some good partition coefficient.
It seems to equilibrate just freely into the brain tissue. The concentrations are a bit lower than in circulation, but we can achieve about tenfold over the EC50 by just IP injection or oral delivery.
And of course, you are continuing the mouse model, and so obviously it will be going to humans. So you mentioned about demyelination. So You think this might have implications for also all the other demyelinating disease, including spinal cord injury and other areas?
I have no idea. You tell me.
Good luck.
Thank you.
So Peter, there was a recent report from Graham Pavitt identifying a new aspect of this whole ISR where the GEF is converted into an inhibitor when the IF2 is phosphorylated. It binds the IF2 and knocks off the initiator tRNA.
Yeah.
And it's not known whether that inhibition uses the dimer or the monomer of the IF2B. So I'm wondering if it's possible that your compound
could influence at that stage.
Yeah, if it has to work as an inhibitor, as a monomer, then that would be another way it could work.
Right. I mean, but what we all have is yeast envy at this stage in the game, because we know so much about these things in yeast, and in mammalian cells here we don't know where this equilibrium lies, for example, right? That in the cell itself, is it pentamer, is it decamer, what would drive these this equilibrium? And there's so much to be done. But what are you doing next week?
So Peter, that compound so potent and specific, is it a natural product or
no? No, it comes out of this out of a publicly available library. It probably was triaged in every screen that has been done before because of its lousy solubility. And I have tried to trace it back, who put it in the library, who synthesized it, why, and it's untraceable as far as.
And if you put a bulky residue or modify one half of the symmetry, will it form an inhibitor of GEF dimerization?
No, half a molecule doesn't work. Or
too low affinity.
So we've not. But these are all things that now, knowing as much as we know, we could go back and screen for again, right, and see whether. All right. Reception time.